Good morning, everybody, or whatever time of day it is for you. I just wanted to hop on here and discuss some of the most important parts of our muscle contraction. So starting out, we're going to cover a few of our muscle tissue cells. We covered a little bit of this in our muscle tissue, or in our tissue chapter, so chapter 5. So I wanted to just remind everybody that we have 3 different types of muscle tissues. We have skeletal, cardiac, and smooth muscle. We have our skeletal muscle. We know that that is the only voluntary type of muscle, so we actually have to think about it. It is consciously controlled. It is multinucleated, meaning it has multiple nuclei, because we know that we need to provide a lot more energy to those skeletal muscles in order to allow for us to do the things that we need to do. And it is striated. This is due to the overlap of our contractile proteins, which we will learn about today. Secondly, we have our cardiac muscle. We know that this is a branched cell. It is uninucleated, only having one nucleus. This is involuntary, but it is also striated. However, it is not striated like our skeletal muscle. It is due to intercalated discs, and this is specifically in the wall of the heart. And our intercalated discs allow easier contractile motion as a whole cell mixed with the next cell so that the heart can pump all at once. And lastly, we have our smooth muscle tissue. These tissue cells are spindle-shaped and they are uninucleated. Once again, we have one nucleus and they are involuntary. So both cardiac and smooth muscle are going to be involuntary, unconsciously controlled, while our skeletal muscle is consciously controlled. Skeletal muscle being attached to bones and used for our motion, while cardiac, heart, and our smooth muscle is mostly in the walls of our internal organs and our digestive tract. I also want to make a reminder that myo is our prefix for muscle. So whenever we see the word myo, we know it has to deal with the muscular system. And that the words muscle fiber and muscle cell can be used in the same way. So if I say muscle fiber, I'm also talking about a muscle cell. While we're talking about our skeletal muscle specifically today, because we're discussing muscle contraction, mostly in our voluntary skeletal muscle, I want to remind you that our skeletal muscle, like our bones, also consist of more than just one type of muscle. Like our bones, how we talked about our bones being the organs of our skeletal system in our skeleton, the bones also have connective tissue, they have blood vessels, they have nervous tissue in the central canal. Our skeletal muscles also have skeletal muscle tissue, nervous tissue, and blood, which we know is a specialized connective tissue, as well as other connective tissues. We will discuss all of that in class. So starting out with our composition of our actual skeletal muscle, I want to talk about this level of organization, because as in every other system that we've discussed, we have a level from smallest to biggest. So starting at the bottom, we have our filaments. These we will discuss in just one moment. So our small contractile filaments, and all of those come together in order to make up a myofibril. So fibrils, kind of like our fibers, muscle fibers all in here, except this is not a full muscle fiber. We take a bunch of myofibrils in order to come together to make a full muscle fiber. So this whole thing is a muscle fiber, and we have multiple muscle fibers in our fascicle. And then our fascicle, we make a bunch of fascicles together, and those make up our muscle. So we're going to start out with our filaments. A bunch of those are going to connect to create a myofibril. A bunch of those are going to come together to make up this muscle fiber. And we can see, it kind of reminds me of the Twizzlers, like the Twizzlers that you can pull apart, because we have all of these. On top of our muscle fiber is going to be our sarcoplasmic reticulum. That's this yellow bit right here, where we are also going to see our nucleus, which we're going to have multiple of because this is our skeletal muscle. It's multinucleated, and these striations are all of our contractile proteins. So once we go to our muscle fiber, this entirety of this picture is one muscle fiber coming together. So we have all of our contractile proteins here. And then in this picture, our sarcoplasmic reticulum is orange. So that's going to be this orange webbing around it, surrounding each myofibril. Our sarcoplasmic reticulum is going to be very important because it holds all of the calcium for our muscle contractions. And so that is going to surround the entirety of the fibril as well as inside every other fibril. And around that sarcoplasmic reticulum, we see these orange bits, these orange rings around it. This is going to be our transverse tubule. Our transverse tubule is what is going to accept some stimulation from nervous tissue and then send it to this myofibril in order for contraction to actually occur. We're going to have a very close relationship between this T tubule, this transverse tubule, and the sarcoplasmic reticulum, because these three pieces, so our cisternae of our sarcoplasmic reticulum and the T tubule together, are going to create something called the triad. So we're going to have sarcoplasmic reticulum, and the T tubule in the middle of it. So we can see this T tubule circling every cisternae of the sarcoplasmic reticulum, but it also incorporates into the other myofibrils. It's not only around one, it continues to sit in all of the other myofibril portions, because once it receives information from nervous tissue, it is going to take that electronic stimulation and work to integrate that information, that signal, to the rest of these contractile proteins. So we can see these openings in the cell membrane because this is one muscle cell. So we can see these openings in these cell membranes, and these are all leading to the T tubule. So if there ever is nervous stimulation that touches down on this cell membrane of this muscle cell, it can reach every single myofibril and allow for muscle contraction. So for our contractile proteins, we're going to have 4 contractile proteins altogether, but our main 2 contractile proteins are going to be called myosin and actin. These are going to be the most important for our contractile proteins and are actually what make these striations. We can see our myosin filaments here in the middle that are a bit of a purple color are going to be called our thick filaments because they are going to be rather thick and also anchor in the middle, while our thin filaments, our actin filaments, are what's going to be pulled along those myosin filaments in order to contract this myofibril. So our myosin filaments are anchored in the middle here by these proteins. We can see these little dots, and that thin actin filaments are going to be pulled along this myosin thread. And that is after we receive neural stimulation through the T tubule, and then calcium is released from the sarcoplasmic reticulum. All right, so let's talk about our muscle fibers. These muscle fibers specifically. So for our muscle contraction, we're going to start from the very, very top. When we want to move a muscle, we have to think it with our brain first, which means that it is going to voluntary contraction, a voluntary muscle, which makes it our skeletal muscle. So we have to start at the top with that nervous stimulation. So here we have the end of a neuron. We talked about the nervous tissue when we were in our tissue chapter. So this is going to be the end of the neuron that is leading to another neuron that will then lead to another one. However, this neuron ends at the muscle. So this is actually going to be our muscle cell down here, this being our muscle cell membrane. And our muscle cell membrane is actually called something special because it works a little bit differently than other muscle cells, than other cells. So this is going to be called our sarcolemma. Sarcolemma. Our sarcolemma is very special because our skeletal muscle cell membrane actually has electrochemical capability. So it is able to receive electrical stimulation from our nervous system and send that as a signal across its cell membrane. So that's why it has a special name, sarcolemma, whatever you prefer to call it. So our sarcolemma is our cell membrane of our muscle tissue, our muscle fiber. So we're going to have our neuron receiving a signal from the brain and sending it down. Our signal is going to be used in yellow here. Signal is coming down. It is going to end up at the end of this end of a neuron, which is also called our synaptic bulb or knob. And that is the entirety of this end of the neuron. At the end of the neuron, it is also going to have a place on the sarcolemma that allows this neuron to meet the muscle, and that is going to be where it actually meets. We have the neuron meeting the muscle, and this we're going to call our neuromuscular junction, or our NMJ, our neuromuscular junction. So that is the actual neuron meeting our muscle tissue. Now the actual canyon here, so this empty space, we see the neuromuscular junction is actually the full end of the nervous tissue meeting the muscle tissue. Now this space in here has a special name as well. This is going to be called our synaptic cleft. Our synaptic cleft is this whole space where there is that opening, where our nervous tissue meets our muscle. When we have both neuron and muscle, it's our neuromuscular junction. The space in between those themselves is called our synaptic cleft. Now, in order to allow a signal to be sent from our nervous tissue, from the synaptic bulbs, the synaptic knob, it has to be able to send something through the synaptic cleft. And for a signal to be sent, we have to start with the chemical messengers of the neuron. And these are going to be called neurotransmitters. And for our nervous tissue specifically, this neurotransmitter is going to be called acetylcholine. ACH, acetylcholine. And this is our neurotransmitter. And this neurotransmitter is what is going to be used for every muscle contraction, especially for skeletal muscle. ACH, acetylcholine, is what's going to be sent from the neuron to the muscle in order to allow a contraction to occur. So starting with acetylcholine, we know that for inside of a cell, because this end of a neuron is the cell of the nervous tissue, the neurotransmitter, this chemical, acetylcholine, is not just going to be sitting by itself, floating around. It has to be held within a certain vessel. And this is going to be our transport vesicles. And we know our transport vesicles, so all this neurotransmitter is sitting inside these transport vesicles. And we know our transport vesicles are made up of the cell membrane. So they have either received the neurotransmitter and created a transport vesicle via endocytosis in order to have those, but then they could also send this neurotransmitter via exocytosis out into the synaptic cleft, and that's actually what is going to occur. But let's go over the rest of our pieces of the cell. So we have our synaptic cleft, we have our acetylcholine and its transport vesicles. In order to send our neurotransmitter across the synaptic cleft to the muscle, we have to have something that will receive that chemical, and that is going to be our receptors. So we're going to have some receptors on this part of the sarcolemma. that are going to sit there and wait for the acetylcholine to send them across the synaptic cleft. Now, this part of our sarcolemma also has a special name. Where our receptors sit waiting for a neurotransmitter, this is going to be called our motor end plate. Motor end plate. You can ignore that. So our motor end plate is going to be what sits right down here. And then we're going to go across. This is still our sarcolemma. So then we have a tube here, so we can guess what this is called. This is going to be our transverse tubule. Transverse tubule, also called our T-tubule. T-tubule. And as we looked at in the slides, this is not what our T-tubule looks like, and it also sits on top of our sarcoplasmic reticulum. But for this picture, in order to draw it out properly, this is how we're going to do it. Just keep in mind that this is not where they sit when we actually look at our muscle fiber. Because next, we are going to have our sarcoplasmic reticulum. So our sarcoplasmic reticulum here is going to hold a lot of our calcium. We know our T-tubule is going to help receive stimulation from the nervous tissue and send it where it needs to go. But once again, remember that our actual muscle fiber, our myofibril looks like this with the striations, and then our sarcoplasmic reticulum is going to encase that myofibril with the T-tubule surrounding it, where we have the cisternae of the sarcoplasmic reticulum, and our T-tubule, where all three of these equal the triad. So our T-tubule is receiving the stimulation from the neuron and then sending it through the sarcoplasmic reticulum to get to the contractile fibers. So once again, keep this in mind, but we're going to draw it out in this manner. So we have our sarcolemma, the cell membrane of the muscle cell. This has electrical capabilities, so it can receive a signal from the nervous tissue. We have our neuromuscular junction, where our synaptic knob or bulb connects with our muscle cell. This being called our neuromuscular junction because we are joining our neuron and our muscle cell. The bit between where our synaptic knob ends and our muscle cell begins is called our synaptic cleft. And then where we're going to receive neurotransmitters across the synaptic cleft is called our motor end plate. If we receive those neurotransmitters, then we're going to continue sending the signal down our sarcolemma and into our T-tubule. So in order to do that, we probably need something to happen up here, right? So we're actually going to make our transport vesicles release the neurotransmitter. We know that that is by a certain transport. So we're going from a concentration, we're going down a concentration gradient, right? So we're actually going to use exocytosis. So once a signal is sent down to the synaptic knob, our transport vesicles get that information and say, hey, we need to travel down. So they're going to travel south to the end of our synaptic knob. Our transport vesicles are going to bind to our cell membrane of our neuron, and they're going to release all of the acetylcholine into our synaptic cleft. So we're going to bind to the cell membrane, release their contents into the synaptic cleft. Once they are in the synaptic cleft, then they can bind to the receptor, bind to the receptor. And once the receptors have acetylcholine bound to them, that tells this muscle cell, oh, we need to start sending this signal. So then this signal travels along the sarcolemma into the transverse tubule. Once the transverse tubule gets that signal, something else is going to occur. So at this point, we have to have that connection between the T-tubule and our sarcoplasmic reticulum. So what we have here is actually a channel. And this channel is going to work by passive diffusion. And we know passive diffusion is where we go from a concentration of a gradient of higher concentration to lower concentration. Remember, because if we fall down the stairs, it takes no effort. We're going to fall down those stairs to lower. So this is going to be our passive diffusion, and that's how this channel is going to work. However, it looks like this channel is closed. So how's it going to change? We know that a change of shape changes the function. So once we send a signal down the T-tubule, that will change the shape of this channel and open it in order to allow whatever is in the sarcoplasmic reticulum to be released. So that's where we are at this point. Let's talk about the sarcoplasmic reticulum. Our sarcoplasmic reticulum is important because it surrounds all of the myofibrils and also is what holds our calcium. So our sarcoplasmic reticulum is going to hold a lot of calcium in this organelle. So if we have a lot of concentration of calcium in our sarcoplasmic reticulum, we've got a lower concentration of calcium outside of the sarcoplasmic reticulum. How do we think it stays in here or gets in here in the first place? Because we do have a channel that when a signal is sent, it opens and then releases that calcium. So we'll put calcium. This is all calcium. So once this channel opens, it's going to release all that calcium. But how does it get there in the first place? We talked about active transport in one of our previous chapters, correct? So what active transport is going to move something from a lower concentration to a higher concentration? At that point, we would need active transport. Go ahead, take a guess, say it out loud. I'll answer. We are going to have a pump. This is going to be a pump. So just like we talked about our sodium-potassium pump, always pumping from a lower concentration to a higher concentration, here we are going to have a calcium pump. This calcium pump is constantly working. It is always taking calcium from the outside, bringing it inside, from the outside, bringing it inside, so that we always have a higher level of calcium inside of our sarcoplasmic reticulum than we do outside. This calcium is going to be very important to allow us to have contractions of the muscle, because once calcium is released, they are going to go and mess with our contractile proteins, our actin and our myosin, in order to allow a contraction to occur. So let's start back at the top. We have our neuron sending a signal of voluntary contraction to the skeletal muscle. So our neuron ends with the synaptic knob or the synaptic bulb. It's going to join with the muscle cell via our neuromuscular junction, and it is going to have that signal be sent down to the end. Once the signal is sent down to the end, that triggers our transport vesicles that have acetylcholine in them to travel south and exit this neuron, this synaptic knob, via exocytosis. They are going to bond with the cell membrane and release the acetylcholine into our synaptic cleft. That acetylcholine is going to transfer across the synaptic cleft and bind with our receptors. Once they have bound with the receptors on the motor end plate, the signal is going to continue down the sarcolemma because the sarcolemma has it is able to send electrical signals. So it is going to continue sending that signal down the cell membrane, the sarcolemma, until it reaches the transverse tubule, our T-tubule. Once the signal reaches the end of the T-tubule, which has our our channel between the T-tubule and the sarcoplasmic reticulum, because the signal is sent, it changes the shape of our channel and allows calcium, because we have a lot of calcium in here, to then be released. And calcium is being released out into the cell to go and do its job. The sarcoplasmic reticulum wants to hoard all of the calcium, so it's going to have a calcium pump that works by active transport to move the calcium from an area of lower concentration to an area of higher concentration inside of the sarcoplasmic reticulum constantly. So even when we do have this channel open and we're allowing all of the calcium to come out, this pump is still acting. It is still working to take all of that escaping calcium and put it back into the sarcoplasmic reticulum. Okay, so let's go through it one more time. We are going to want to move our finger. So we need to send a signal from our brain down our neuron until it reaches the synaptic knob. Once that signal is sent to the end, it triggers our acetylcholine to reach the end of our synaptic knob and then exit via exocytosis. This is then going to have acetylcholine released into our synaptic cleft. Once acetylcholine is released into the synaptic cleft, it has to travel across and then bind with receptors that sit on our motor end plate. Once acetylcholine binds to these receptors, the signal is going to continue to be sent along the sarcolemma. It is going to reach our T-tubule, and then it is going to change the shape of this channel and open the channel to allow all of the calcium that was inside of our sarcoplasmic reticulum to now be released. But we know that the sarcoplasmic reticulum also has a pump. This is a calcium pump because it wants to continue to take all of the calcium from this outer part of the cell back into the sarcoplasmic reticulum, constantly working against any of our calcium being released. All right, so our calcium is released into the cell. What comes next? We are going to have our calcium travel over. We're going to ignore that I used the same color for calcium as I did for one of our troponin molecules. So we're going to have the darker blue be calcium. So let's say the calcium is now released into the cell. It's coming over in order to go to our contractile proteins. Now, before we get to that, I need to introduce our two other contractile proteins because we talked about myosin and we talked about actin. Now, this is the picture that we saw in the slides, with myosin being a thick filament that is anchored in the middle and actin being our thin filament that is going to be pulled towards the centers. So what it actually looks like. We have myosin, and myosin has a very thick middle band, but it also has all of these small pieces, these small proteins that come off of the side. So these are going to be our myosin heads that come off the top and the bottom in a very circular movement. It's kind of like you have a bunch of or a circle in the middle and then a bunch of points coming off of it. So our myosin head, even though we draw it like this, there is also myosin heads coming off here, coming off there, coming off here. Because if we take our myofibril and we cut a cross section of it, where we look down the cut, this is going to be what it looks like. We have our thick myosin proteins in pink. We're going to have a circle of actin all around them. So it doesn't just look like one myosin to one actin. We're going to have one myosin with probably 6 to as many as we can, actin strands around it as well. So our myosin thread is going to sit there with the myosin heads waiting, while our actin is going to sit all around our myosin threads hanging out, and our actin is going to have a site on it that's going to be called its active site that our myosin head wants to bind to. So all these myosin heads are sitting there waiting for actin to be available for the myosin head to bind and pull these actin threads towards the middle. But the actin threads are not always, or the active sites on actin are not always available, which is why we don't have constant contractions in our body when we don't actually want it. That's where our troponin and our tropomyosin are going to come in. So if we have our myosin thread here, we have our myosin heads coming off of the top. We have our purple actin thread. Our tropomyosin is going to be a roping protein that circles around our actin. And our troponin is going to be a small circular protein that is attached to tropomyosin, which is why they have very similar names. So tropomyosin is the roping green protein around actin, while troponin is going to sit on tropomyosin. So here's the fun part. Our troponin is going to change shape when calcium binds to it. So really what had to happen is the neuron had to send a signal. It had to reach the muscle cell. It had to travel across the sarcolemma into the T-tubule. Here, hold on, I'll look at it this way. The signal had to be sent. Acetylcholine had to travel across, bind to a receptor. The signal had to continue into the T-tubule, change the shape to allow the calcium to be released from this channel, and then the calcium leaves into the cell in order to come over to these contractile proteins. And for calcium to come and bind to troponin. So that is calcium's job. It wants to come and bind to troponin so that we can move tropomyosin away from actin and allow the active site to be available so that the myosin head can then bind into the active site and pull our actin towards the middle. So we have calcium. It's released from the sarcoplasmic reticulum. It's coming over, it is going to bind to troponin, and then it is going to lift troponin off of tropomyosin, because as we know, when something binds to a protein, it is going to change its shape. And once we change the shape, we change the function. So instead of being the gatekeeper of actin's active sites, now troponin has calcium bound to it. So calcium is going to come bind to troponin. And once it does, troponin is going to change its shape and then pull tropomyosin, because we still have calcium on here, is going to pull tropomyosin off of actin, off of the active sites. So now the myosin head can bind. So we have now, this doesn't make another head, it's just actually reaching up and then binding to the active site to allow you to pull. All of these myosin heads are going to bind and pull the actin towards the center so that we can have that contraction of the muscle. That contractile protein is going to do its job and pull the actin towards the middle of the cell where myosin is bound. So I have an analogy that might help with this. We are going to have somebody who really wants to ride their bike. They really want to ride their bike. So here we're going to have, sorry, I need to change colors. So our bike analogy. We really want to ride our bike. Our bike just so happens to be actin. So actin is our bike. And you really, really want to ride your bike. You are myosin. You are myosin, and you really want to ride your bike because you just need your foot in order to reach the pedal. However, you can't push the pedal because there happens to be something blocking you. At that point, what do we have around the bike? Probably a chain and a lock that doesn't allow you to put your pedal on the bike and ride it. There's something in the way, probably through the tire, not allowing you to reach the active site and to make that move happen. So with that, we're going to have tropomyosin, our roping protein. That is tropomyosin is our chain that is going to be looped all through the wheel, and you can't move the wheel. And in order to move that chain, you have to unlock the lock. Oh, I did that in the wrong way. That's fine. Troponin is going to be our lock. So if you, as myosin, really want to ride your bike, you just want to put your foot on that pedal, on the active site, and move it and ride it, you can't until you have unlocked this lock. Now can we take a guess as to what our lock is going to be? Say it out loud, even though I can't hear you. Beautiful. It's going to be our calcium. So calcium is going to be the key. So we can't have a muscle contract until we have unlocked that bike chain. We can't have movement of that bike until we take the chain off of it. And we can't take the chain off of it until we change the shape of that lock with our key and then have it be taken off. So once we have the key, once calcium has been released from our sarcoplasmic reticulum, we are able to send that to troponin. We are going to use calcium as the lock, change the shape of it, and then troponin is going to pull that chain, pull tropomyosin off of actin. Actin being your bike, and now finally, you, as myosin, can put your foot on that pedal and move it, and go on a happy little bike ride if you would like. All right, so let's go over this just one last time. For our muscle contraction, this is skeletal muscle, so it is voluntary. First, you have to have a signal sent from your brain down the neuron. It has to reach our synaptic knob or bulb at the very end. Once that signal is released, is sent and received at the end, acetylcholine is triggered to go south to the end of the synaptic bulb and release the neurotransmitter acetylcholine via exocytosis. Acetylcholine is going to be sent across the synaptic cleft and then bind with receptors. These receptors are situated on our motor end plate of our muscle cell membrane, of our sarcolemma. So once acetylcholine has been received from our receptors, the signal is going to continue to be sent down the sarcolemma and into the T-tubule. The T-tubule receiving that signal is then going to change the shape of this channel and open to release calcium from the sarcoplasmic reticulum. Now our calcium is going to constantly be pumping via active transport calcium from the low concentration in the cell back into the sarcoplasmic reticulum. So once that channel is opened via passive diffusion from high to low, we are going to send all of this calcium out into the cell. And once that calcium leaves into the cell, it is then going to go and find some contractile proteins. It is going to be sent to bind with troponin that is on tropomyosin. It is going to change the shape of troponin, which then lifts troponin and tropomyosin off of actin. This allows the active sites on actin for myosin to bind and to pull in order to have the muscle shrink, to contract, and a proper contraction has now occurred. And that's all I have for you guys. I hope this clears some things up and is a really good starting place. I will see you all on Tuesday.